Welcome back. So we're really entering, well, I don't know. The whole course is fun to me, but we're really entering a part of the course that I find to be delightful, and that's the subject of positional information, but more generally, how we bring together the ideas that we already introduced about temporal evolution with spatial evolution. And I mean, we already got a hint of that in the context of diffusion, where we saw that we can have molecules and they can move in position, not only change the concentration over time at a given point because of chemistry, but also could change their position and change concentration through that means. So what this particular vignette is about is it's really an introduction. I'm not going to do any math. I'm going to basically provide the backdrop, and then in the coming vignettes we'll look into this stuff more mathematically and sort of try to work out what the implications are of this, let's call it pathetic thinking. So I think it was only last week that Louis Wolpert left this life after a long and productive life. And in 1969, he wrote this very seminal paper called Positional Information in the Spatial Pattern of Cellular Differentiation. And, you know, this is, it reminds me a bit of the quip about Mark Twain and classics, and I'm not going to remember the words exactly, but, you know, he said something along the lines that a classic is something that everybody talks about, but nobody's read. And what I can say here is that James Sharp, for the 50th anniversary of this paper, wrote a really nice little position piece about the positional information, the French flag model, and I appreciated it very much because it calls attention to the way that we caricature things, the way that we think we understand what somebody was thinking, and you know, this comes out in all sorts of different ways, in the way we judge what somebody else means or is or thinks or whatever, and we really don't have it right because we're interested in the soundbite. So, you know, the soundbite on Wolpert is that he presented the French flag model, which, you know, that's not actually true. There was a paper in a book the year before this, a really interesting book on theoretical biology, which I have on my Kindle, that was edited by Waddington, and in it, Wolpert identified what he called the French flag problem. And I will show you a little bit about that in the coming vignettes. But at any rate, so this paper introduced, let me just read you from the abstract. Pattern formation thus differs from molecular differentiation, which is mainly concerned with the control of synthesis of specific macromolecules within cells rather than the spatial arrangement of the cells. And, you know, we've just gone through all of that with respect to Jacob and Monod and the modern theory of gene regulation. We definitely looked at the way that that's a nice way to put it, molecular differentiation. It is suggested that there may be a universal mechanism whereby the translation of genetic information into spatial patterns of differentiation is achieved. Beautiful, you know, it's a hypothesis. And, you know, people love to say, well, biology's full of exceptions. Fine, biology's full of exceptions. That doesn't mean that we shouldn't think big and try to come up with hypotheses and see how far they take us. The basis of this is a mechanism whereby the cells in a developing system may have their position specified with respect to one or more points in the system. This specification of position is positional information. And so we will know that for the purposes of what I'm going to do in this course in terms of ideas like the so-called Morphogen Gradient. So this is experiments from Thomas Greger's lab at Princeton, and what's being shown is the bicoid concentration is a function of length along the embryo. The left-hand side is the anterior end, the right-hand side is the posterior end, so you can see that there's higher concentration. I'll talk about this, but the mother leaves behind some mRNA that's localized on the anterior end. It's produced, it diffuses away, but has a finite lifetime for degradation. And those two facts together suffice to provide what amounts to a morphogen gradient. That morphogen gradient has a particular functional form, and we will calculate that functional form. So that's the morphogen, and then the sort of thing that Wolpert had in mind with respect to the French flag problem was this idea that there would be some thresholds. And if the level of morphogen was above threshold 1, as you see here, so that's in this region, then that means you'll suffer the blue fate. If you're between threshold 1 and threshold 2, you know, which is this white region, then that will lead to fate white. And then if you're over here, which is low concentration of morphogen, that will lead to fate red. That's the kind of idea. And so I just wanted to comment, you know, that people have done really interesting experiments many people. There's a beautiful experiment from Nusslein, Volhard, and Driver, but also Steve Small has been doing really wonderful things. And the way I would put it is, in the language of synthetic biology, is that they've been reprogramming the gradient. And then on the basis of that reprogrammed gradient, trying to understand the nature of the resulting phenotype. What does the embryo develop into? So I just wanted, for fun, to comment on a few things. So first of all, we've now touched on the fact that Wolpert had this idea of positional information, and I will contrast that, as do people like Sharp and Briscoe and others, with the Turing type idea. And when I say positional information, I'm going to be thinking more generally than the original Wolpert paper. And when I say Turing, I'm going to be thinking more generally than the original Turing paper. In the same way that when I say allostery, I'm going to be thinking more generally than the original MWC model. I'm not going to get into the debate about MWC versus KNF. These are two models that were both developed in the 60s, around which was swirling tons of passion. But for me, they're both homework exercises in allostery, and I don't, we can argue about the details, but the notion of allostery is the general thing. Here, the notion of positional information is the general point, and there's many different mathematical realizations, and I don't know whether it's worth getting all hot about those specifics. So ditto on Turing. We're going to come back to that. So what I wanted to say is that, as is the case in many occasions in the rise of modern molecular biology, often Crick was there. So this is a paper from roughly 1970, and what I hope that those of you that are following the course will get out of it, I think it's really worth your while to look at. And what I hope that you'll get out of it is, in paging through it, you'll see that he has written down all sorts of things like we've done in this course. In fact, this paper is his order of magnitude paper, is the way I'd put it. So he has expressions such as time is equal to L squared over D, and indeed, you can see that the time to set up a morphogen gradient is like if I'm on the desert island with Wilson and you ask me for an instantaneous 5-second answer, the answer is length of the embryo squared divided by the diffusion coefficient. That's the time scale. Similarly, if I want to know how long it's going to take to cook a turkey at Thanksgiving, the way I'll think that one through is I'll figure out there's something called the thermal diffusivity. So I'll take the radius of the turkey squared, divide by the thermal diffusivity, and that gives me an estimate of how long it takes to cook the turkey. Anyway, so what's my point with respect to this particular paper? It's just to say that Crick had, I want you to appreciate it as an order of magnitude reasoning type of paper. That was followed up by this paper, which is really fun. The time needed to set up a gradient, detailed calculations, and for those of you following the course, I just am getting ready to hand out homework number 4, and in homework number 4, we work out FRAP in a one-dimensional situation, and what you will find is that the mathematics that you're going to exploit is more or less identical to what you see here as equation 2. The way I formulated the problem and handed it out, you'll see that C of x, which is the concentration, and time is given by this sort of expression in terms of signs, and the thing that I guess I want to point out is that the expression has an e to the minus n squared pi squared capital T, and notice that Crick, a few lines down, says we have expressed time in a convenient dimensionless form by putting T over L squared over D. So he's dividing by this diffusion time, and that's the natural time scale for this problem. So at any rate, this is a cool paper to take a look at, and what I think you'll find is you'll appreciate the shared culture. Here Crick himself is saying, look, if you want to know about all this stuff, go look up the book Conduction of Heat in Solids and the Mathematics of Diffusion. That's exactly right. I have both of those books sitting on my shelf at home. And so there's this shared culture that really goes all the way back to Fourier. And there too, I can show you, and maybe I will try to remember, I will show you his early 1800s theory of heat, and in it you will see exactly the kinds of expressions that Crick used right here, which is the introduction to Fourier series. Very, very beautiful, and provocative and powerful. It's a superpower, because once you know how to do this, at least for certain classes of problems, you can get a long way. And then if you allow yourself the generalization to Fourier transforms as opposed to Fourier series, you can get even farther. Really, really far with those kinds of ideas. So the summary here is that I wanted to talk about positional information. That's going to serve as the backdrop for the coming few vignettes, and our treatment of positional information is going to really focus on the ideas set forth by Wolpert and the math that goes with them, and then the experiments in the spirit of closing the loop that go with them. And then we're going to compare and contrast, weigh that against a totally different view in which there is not a coordinate system that's set up. And that's the Turing mechanism where basically if you like, the coordinate system, the lattice actually arises spontaneously. And the reason that this kind of thing is important biologically is there are lots of examples where you look at a biological system and you realize that there's something sophisticated going on. If you look at how well bacteria find their middle, the answer is they find their middles very well. If you look at this thing from Nusslein-Volhard, which I show you here, the cephalic furrow is well positioned depending upon the dosage of the bicoid gene, and that actually was a super critical, smart experiment to try and challenge the Wolpert idea, to try and find out whether the cephalic furrow, this position, shifted in response to changing the nature of the gradient. And indeed it did, although as we will see, it doesn't work out quantitatively quite as well as one would hope. And that opens the door for more thinking. So that's another example. Then there are other interesting things like why do we end up having 5 digits, or why are my 2 legs nearly the same length, and what are the biological mechanisms that impose that kind of spatial ordering. And so all of this falls within the rubric of the general question of how do we unite our thinking about time, which we've spent an exorbitant amount of time on, and space, which we started in the context of diffusion, and we're going to spend a lot more time thinking about from here on in the context of active matter and other things. So the point is we've basically launched ourselves into these space-time problems, and really what we're interested in, and this could serve as a siren call for much of modern science, whether it's in thinking about geology or astrophysics or whatever, which is we want to know the space-time evolution of the systems that we're interested in, and that's what we're now going to turn to.